I am Hilary Robbins. I'm an epidemiologist at the International Agency for Research on Cancer, which is the specialized cancer research agency of the WHO in Lyon, France. And the project that I have presented at the World Conference on Lung Cancer is comparing lung cancer detection between the Manchester lung health checks and the US National Lung Screening Trial. So in the Manchester lung health checks, this was a pilot study of lung cancer screening carried out in a largely lower income population of Manchester in the UK. And over 2 screens spaced 1 year apart, the lung cancer detection rate was 4.5%. So 4.5% of the screen participants had a lung cancer detected. Comparing this with the National Lung Screening Trial. So the US National Lung Screening Trial had 3 screens spaced 1 year apart. But if we consider only the first 2 screens spaced 1 year apart, so the same design as Manchester, the lung cancer detection rate was 1.7%, so quite a bit lower. And we wanted to understand essentially why this was. So there's a few different reasons concretely that we know could have played a part. The first is that the Manchester study used risk model-based eligibility. So in order to be eligible for the Manchester study, you had to have 6-year lung cancer risk over 1.51%, as predicted by the PLCO M2012 risk model. Whereas for the NLST, they used criteria based on age and pack years. So age 55 to 74, at least 30 pack years, and no more than 15 quit years. And these eligibility criteria in the NLST do allow lower risk people to come in to this study as compared to Manchester. So the first thing that we did is we imposed the risk-based eligibility on the NLST study. So we forced the eligibility criteria to be the same as they were in Manchester by basically excluding the people in the NLST who would not have been eligible for Manchester. And then we compared the detection rates again. And we found that the detection rate in the Manchester eligible portion of the NLST was 2.1%. So whereas before we had an increase of 2.6-fold, the increase reduced to 2.1-fold. So it does appear that this influence was playing a part. Another concrete influence that we know could be contributing to the difference is that even after applying the same floor of risk for the eligibility, so saying you had to have higher than 1.51% risk to be eligible, the distributions of risk above that threshold could be different. And we found that this was indeed the case. So the distribution of risk in the Manchester participants was shifted towards higher risks than in the NLST. So essentially you can think of it as the mean risk being quite a bit higher in Manchester than in the NLST. So we subsequently used standardization to remove this influence, to basically impose the same distribution of risk on both populations. And this further reduced that increase in the detection rate to 1.7-fold. So we started with 2.6-fold. We imposed the same eligibility threshold. We got 2.1-fold increase. And then we imposed the same risk distribution and we got a 1.7-fold increase. So what we see here is that these two sort of eligibility kind of epidemiologic influences are certainly playing a big part and why there was higher lung cancer detection in Manchester than in the NLST, but there is some residual increase. And what we did is we looked at this increase across different subgroups of Manchester to see whether it was really happening in particular types of people and not in other particular types of people. And the most interesting finding there was that the increase was highest in groups with lower socioeconomic status. The increased detection relative to the NLST was very strong in people with lower socioeconomic status and was not statistically significant in groups with the highest socioeconomic status. And it's worth mentioning that in Manchester, the whole study population was relatively low income. So if we were screening a higher income population in the UK, we might not expect to see any difference. So this points out that socioeconomic status is potentially an important driver of lung cancer detection, but if it's not captured appropriately in the risk prediction models that are used for eligibility, then it could lead to important disparities.